Hey, good afternoon. It's a pleasure to follow 40MT, who's one of our partners. Hope everyone's enjoying the meeting and enjoying the lovely weather. Pleased to tell you a little bit about Arbor. So Arbor was co-founded about 7 years ago by Feng Zhang, who's the CRISPR pioneer out of the Broad, and David Walt, who is out of Harvard and the Wyss Institute. David had co-founded a company quite a few of us use, Illumina. And they got together and founded the company on an idea as opposed to IP. And that idea was there's likely to be a tremendous amount of interesting editing material that evolution has created. And why don't we go identify it, harness it, and identify a toolbox of approaches for editing. Now, if you flash forward, we now have a pretty expansive toolbox of proprietary editors. And importantly, it opens up a lot of new targets because we are focused on very small editing approaches, which allow us to move out of the liver. But having a toolbox doesn't do us any good if we don't have a focus on translating these into therapies. We do have a goal of 3 clinical filings over the next 3 years. Our first one is targeted actually for later this year. And we are also have our next couple programs will be in the CNS space. Importantly, it's important that we have the right investor base. We have a very strong cadre of investors. You can see some of them listed here, as well as a strong group of partners. And the other, I think, important element as we think about a company, you need to have good technology, good people, and a great strategy. And we have really talented folks that do understand how to turn these novel technologies into therapeutics. And so as you can see here, some of our talented team, we have over 20 IND and CTA filings of experience in much of that in these advanced therapeutics, and that's really important. In addition, our founders are very involved in the company. Feng chairs our scientific advisory board, and David is on our board. So we've got really strong, talented folks. The question then is, on the technology, why is it different? Is it different? I think what's important, if we take a look at Cas9, which is an important workhorse, I think, for across our industry and broadly in academia. And it is, it's been around a long time. Many people are very comfortable with it. I think what's important from our perspective is one needs a variety of editing approaches beyond Cas9. Why? Cas9 usually has a single cut type. The length of the edit is very defined. It's usually a +1 indel, which means it inserts one nucleotide. It has a restricted PAM. So the PAM, think of it as almost like the, if you have you write a letter, which I don't think anyone writes letters anymore, but you write the address, and your city, you write a city-state zip code. The PAM is a lot like a city or state. It tells the nuclease where it could bind. And that's important because if you only have a single PAM, you're only going to be able to go to a certain city and state. But if you have multiple PAMs, you can bind to anywhere on the genome. The guide is what then tells the nuclease where it must bind and what type of editing it's going to do. So one advantage that we do have is we have much smaller guides. And this is important as you move to more sophisticated editing because they're a lot easier to make and manufacture. In addition, there's a lot of different editing approaches one can undertake in this space. And I think that's a knockdown that is utilized by some folks is really important, and it provides benefit in many diseases. There's also other approaches you can take to excise or remove large pieces of DNA where you may have some kind of repeat disease. Other approaches where you may need to rewrite individual mutations, or finally you may want to do insertions. And so having a broad base of these effectors allows us to really target effectively any target or disease in the genome. Now that's wonderful, but we can't do it all. And so we have to think about where and how do we focus. So our original focus has been on a lead indication where we can utilize existing biology, utilize de-risked delivery, and also identify how do we have a clear clinical and regulatory path. So our lead indications in primary hyperoxaluria. The last and most important piece for us on indication selection is we have to be first from an editing perspective. If we can't be first, then we will not go there. And so with this first program in the liver, our subsequent programs in the liver are going to be in the RT editing space, again, in diseases where we can be first. Outside of the liver, which I think is really the next big horizon for editing, we're moving into the CNS. ALS is a great starter space for that move into the CNS for a couple reasons. One, it's a very defined space. There are delivery approaches that you can access spinal motor neurons at high levels of transduction. And in addition, ALS, as all of us know, is a horrible disease with effectively no treatments unless you have one specific mutation. So this allows us to really begin to expand gene editing beyond the liver. One important piece for us has been focus. We have a fairly small pipeline, 5 programs we're focusing on. Goal to have 3 of these into the clinic over the next 3 years. And this is important, again, from a focus perspective and a capital efficiency. And this is something we've been very thoughtful about. We don't want to work on too many things because then everything moves slowly. You can see we have 3 programs targeting ALS, both the sporadic and the genetic patients. And then we do have multiple collaborations. One thing we are not doing as a company is ex vivo cell therapy. That is a very different capital and company structure than an in vivo therapeutic approach. And so we do partner in that space. We partner, give folks access to our technology that's unencumbered, and they can do the particular cell therapy they need to do. And we have collaborations with Edagene, Allogene, and Vertex in that space. Delivery is an important piece of the puzzle for editing. And we have partnered on the LNP side and the AV side with folks who have the next generation approaches that allow us to get to the right tissue. Our view is that we do not have to be creating our own delivery modality. And the reason for that is there are a lot of companies who have spent a lot of capital building differentiated delivery approaches that we can then partner with to bring a differentiated cargo. And we believe that there is no one-size-fits-all for delivery. It depends on the tissue, depends on the patient, depends on the disease. And so by having a much more agnostic approach allows us to pick and choose the right and best partner for a particular disease. So I'll cover quickly our lead indication, primary hyperoxaluria. Simply, this disease is a disease where there's a mutation in a particular gene which leads to a vast overproduction of oxalate. Now, oxalate is a water-insoluble compound, which is always a bad thing in the body because it comes out. It precipitates out in the kidney to form stones. And most people think of kidney stones as little round pebbles that come out and, it's pain's worse than childbirth. I've never had them, but never want to. In this particular case, the patients often describe them as sea urchins. They're spiny and really nasty-looking stones, and they're often diagnosed as toddlers. So if you can imagine having a toddler trying to pass a kidney stone that's shaped like a sea urchin, it's not a pleasant process. These patients often, by the time they are in their early adulthood, have had a kidney transplant or are on dialysis. So it's a very devastating disease. The interesting piece is it emanates from a healthy liver, but destroys the kidneys. Our approach is to impact this disease by basically eliminating HAO1. So HAO1 is an important enzyme that results in formation of a downstream cascade, which leads to oxalate. By blocking this enzyme, we can shift the cascade up to what's called glycolate. Glycolate is water soluble, and it can be passed through the kidneys so that you can prevent the buildup of the oxalate in the kidneys. There is an approved therapeutic from Alnylam called Oxlumo, and they have done a fantastic job of building the market. And one of the toughest things going into a new orphan disease is building the market. And so in this case, Alnylam's done a fantastic job of that, and we're hoping to continue to help build off of the strength that they have. There are between 1,000 and 2,000 patients in the US and EU, and we know there's a significant higher number of patients in the rest of the world, particularly in the Middle East and down through I think the Levant region, as it's called. And so there's a significant number of patients here. About 500 are currently on Oxlumo, and Alnylam continues to grow that patient population quarterly. So just a couple of data slides. This is looking at a time course experiment of editing over time versus protein expression of the protein we're eliminating and the impact on the biomarker. So in the perfect world, the red is editing over time. The way it's measured in gene editing is called indels. So you grind up the liver, you sequence a gazillion cells and determine what % of the cells have the edit you need. And in the particular LNP we're using from Acuitas targets the hepatocytes. Now, the hepatocytes make up about 60 to 70% of the liver, depending on the particular mouse strain and or species, if you're going mice versus non-human primates. So you can see here in red, we begin to see editing very, very quickly with the LNP, delivering the mRNA and the guide, and that really tops out within 1 to 2 days. You can see the protein in blue drops like a rock, as you would expect. And then in the right-hand side, you begin to see that glycolate levels go up significantly, which is exactly recapitulates the biology we want to see and allows us to really think about how to model for the NHP studies and beyond. So we moved into non-human primates, went through a series of optimizations, selecting the right LNP formulation, the right guide, and finally in the far right, we knew in the primates based on our data and Alnylam's data, we need to meet or exceed 55% indels in that NHP liver. We averaged about 60% indels. And importantly, the tolerability and safety was very good. And that's really important because your two risks for gene editing are on your acute delivery and then any sort of off targets. And we've certainly, our team's done a good job of de-risking both of those elements. We have had pre-IND and pre-CTA meetings across Europe and in the US, and the target filing by end of year. So quickly touch on ALS. ALS, 90% of the patients are actually sporadic, which is terrifying to me as a target patient, which is a white male that's over the age of 50, because it strikes people in their prime, and it's devastating when it hits. 10% of the patients are genetic. They have genetic drivers. The biology has been greatly understood for the sporadic patients over the last few years, so we're beginning to identify targets that can be utilized for gene editing across the sporadic patients, as well as the opportunity to target the genetic patients. So I'll just cover quickly one approach we're taking, which is targeting a protein called stathmin-2. It is a microtubule binding protein. It's one of the most abundant proteins in the axon, and it plays a key role in axonal health. When you lose or you have a dysfunctional stathmin-2, the axon degenerates and you lose your neuron. The reason for this in the sporadic patients is you actually uncover a cryptic splice site, so you get a truncated protein due to the missplicing. So what we've done is utilize our technology. We have a particular nuclease that when it cuts, it can cut between 10 and 20 nucleotides in one shot. That's perfect for mapping and identifying a cryptic splice site and removing it. And so here you can see some of the data in the top on in vitro. We can, you look at the beautiful human patient-derived neurons. On the control, beautiful processes coming out. When you have the truncated protein, they're gone. When we restore the full-length protein, the process is restored. So it's a really nice demonstration of the biology. When we moved into a mouse model, we see the exact same thing. We're able to, using the AAV, we can greatly decrease that truncated protein and greatly increase the full length. This program is now in non-human primates. So I'll skip quickly to partners, because the little thing's beginning to flash yellow on me. And we have partners are important. We have 3 partnerships with Vertex. Vertex has come back several times to expand our partnerships. The 4DMT partnership's important, and our ex vivo partners are also extremely important for us. And we continue to partner not only on the ex vivo side, giving folks access to an unfettered technology, but also on the in vivo side. So I'll pause there. We are a next generation editing company. We've got significant catalysts over the coming couple of years, and we're really excited to see the first non-Cas9 enter the clinic over the coming months. So thank you.